We have been undertaking the effort of developing novel mouse models of Richter syndrome, actually with the intention initially of modeling chronic lymphocytic leukemia. We introduced loss of function driver mutations in animals through CRISPR-Cas9 gene editing. And some of these lesions were key to actually the transformation into Richter syndrome, which represents one of the foremost clinical challenges nowadays, with 2 to 10% of CLL patients transforming into DLBCL-like histology, which is almost invariably fatal and with very limited therapeutic options. And through these mouse models, we were able to highlight key pathways that are involved in transformed disease and key genetic drivers that are responsible for transformation. And these include, of course, the very notable TP53 mutation, which is frequently occurring in high-risk CLL, but also in Richter syndrome patients. And importantly, also mutation in MGA, which is a MYC regulator, and CHD2, which is a chromatin remodeler. And we demonstrate through a number of assays and multi-omic analysis, particularly transcriptional, epigenetic, and genomic analysis, we demonstrate that a key pathway that is responsible for transformation is aberrant MYC signaling, similar to other types of lymphomas. And this, in turn, leads to vulnerability to MYC inhibitors, but also mTOR inhibitors, and also PI3 kinase inhibitors. And so, whereas quite unexpectedly so, we saw a key diminished interferon gamma signaling signature that is unique to Richter's, and especially to these models. We are right now in the process of trying to validate some of these dependencies in human samples to try and understand better how faithful these models are to human disease. But they certainly represented a key innovation, as they are uniquely able to recapitulate transformation from indolent CLL, which is something that it's uniquely affordable in these platforms, and not so much so in PDX and xenografts that are instead based on transplantation of primary samples into immunodeficient recipients. And we are, of course, in the process of further understanding functional dependencies and therapeutic vulnerabilities, also in the context of checkpoint blockade therapy, which seems to be more effective in Richter's patients as opposed to CLL. We still don't understand why, and we're right now in the process of understanding it through both the modeling and, of course, other efforts in the lab are focused on the human analysis. And we do think that these studies will really allow to highlight key functional pathways that can be then targeted for therapy, and provide models that uniquely allow to study therapeutic options and to evaluate even the effect of single agents or combinatorial treatments in vivo.